These notes are going to be on cell transport, or how things move in and out of the cell membrane. So there's two types. You have passive transport, which doesn't use energy, and active transport, which does. So first we have passive transport. There are three different types: diffusion, facilitated diffusion, and osmosis. The first one is diffusion. So this is the movement of particles from high concentration to low concentration. It's going to move lipids, oxygen, and carbon dioxide. So diffusion is working towards equilibrium or homeostasis, which you hopefully know. This is moving just through the membrane. So if you look at this diagram over here, there's nothing helping it. It's just floating in between the heads and tails of the cell membrane. Next, we have facilitated diffusion. Facilitated means it has a helper. So in this case, it's a protein. So this is the movement of materials across the membrane using proteins. Basically channel proteins, which is going to move things like glucose, which are all these little dots here. Glucose is too large to fit through the cell membrane this way, so it has to use the proteins. The last type of passive transport is osmosis. This is only water. Don't get this confused. Osmosis is water. I think about water's H2O. Osmosis starts with O. Easy way to remember it. This is where it's going from high to low. So we drew a picture down a hill, which I'll draw at the end here. But if you're going up the hill, you're using energy. Down the hill, you're not. So high to low would be downhill. So all of passive transport is downhill. Osmosis can also result in three types of solutions. Remember, we are just talking about the movement of water in these types of solutions. So you have iso, hypo, hyper. And we'll talk about each one of those in a minute. So isotonic is when they're moving in and out at the same rate. So it's going to be this normal looking cell here. And this is an animal cell, or actually a red blood cell. So red blood cells want to look like this. So animal cells in general want to be isotonic. So animal cells are going to do best in an isotonic solution. Next we have hypotonic. So this is where the solute concentration is higher inside of the cell, meaning the solute is higher, so the water percent is lower. So water's going into the cell because it wants to equal out. So this is your swollen up cell that's ready to explode if it's an animal cell. Animal only. Now, down here, important to note, plant cells want to be hypotonic. This allows them to stand up tall. So if you forget to water your plants, their cells are actually becoming isotonic or even hypertonic. So plant cells want to be hypotonic. And this constant pressure on the cell walls, because the cell walls prevent them from bursting, is called turgor pressure. That's a word you will see on your test. Last up, we have hypertonic solution. So this is where the solute concentration is higher outside of the cell. So this would be maybe putting a cell in salt water, because you have much more salt outside than inside. So water is going to flow out of the cell, making it shriveled. Okay, it's going to shrink up and become all shrivelly. No cells want to be hypertonic. So now we have active transport. So this is the movement of particles across the membrane, very important, using energy. They're either pushed in or pulled out, and there's three types. So you have transport proteins, endocytosis, exocytosis. This is exo. Oh, I'm sorry. Oh, forget I wrote that. This is endo because it's being taken in. So this is endo. This is exo because it's exiting. So let's go over those. So first of all, with transport proteins, these are what I called in class the Pac-Man proteins. So these are the ones that need energy. And what's going to happen is there's this little starburst of ATP, which is going to come hit this cell to make it function. So it's using a carrier protein to help the particles travel through. So it's going to go through. This is like step by step here. Next we have endocytosis. If you remember, endo means in or enter. So this is when the cell surrounds and takes in a particle. Another word you might see is engulfing. This is kind of how some cells eat. Certain one-celled organisms, this is actually how they get their food. And exocytosis is out or exit. So this is how you get things out of the membrane. So this is how a cell would get rid of waste. So big picture. So your hydrophobic molecules, which are your gases, can just go straight through. Water can also just go straight through. Okay? Things like glucose, sucrose, which are sugars, and ions need a protein to work. So you'll see them in this picture turning away because they can't fit through the membrane. So just remember, the smallest stuff can fit through, the larger stuff needs a protein. And then last here, I'm going to draw this mountain that we drew in class. So if you have a mountain and you're going to bike up it, forgive my drawing here, you're going to bike uphill, right? This is low, this is high, and of course the other end is low. So, low to high is going to use energy, right? If you're going to pedal up a hill, you're going to be tired when you get to the top. Now, high to low doesn't need energy. You're just going to coast down the hill. You don't even have to pedal if you don't want to. So the high to low is like your passive transport. And the low to high is like your active transport. So low to high uses energy just like you would to climb a hill, and it's the same thing as active transport. High to low uses no energy, and it's passive transport. And this high and low, think about it as the concentration gradient. So this hill is similar to your concentration gradient. And this is something you will also have to know how to do for your test.